The first phylum of animal kingdom belonging to the non-chordates is phylum Porifera. Let us dissect the name Porifera. It is quite clear from the name that something about pores is to be discussed. So, Porifera would give us the term pores, and when we talk about pores, this phylum is otherwise in common terms known as the phylum of sponges. Now, before going to sponges, just recall what a sponge is like. There are many pores inside it. Same way, the organism, an animal is going to have pores. Such would be the arrangement of the cells inside that animal, that there would be pores that would be present, and through that pores and some cavity that could be present in the animal's body plan, we are going to see how it performs its body functions. What we have to see is the characteristic features that this phylum's animals are going to have. Okay. Now, first thing, in any phylum, whichever we are going to study now, you have to remember the things which are mentioned over here after understanding them. The first thing is that they are primitive. That means they are not highly advanced. They are multicellular. It is not going to be a single celled sponge ever. They would be multicellular, because whatever we have read till now, we did see that there was unicellular fungi, some algae were unicellular, but in the case of animals over here, we are going to see the multicellular animals which are primitive, and they belong to phylum Porifera, in which we have pores. Now, these primitive animals, they are primitive in the sense that their organization is nothing beyond cell aggregate. Try to recall what we read in fungi and what we read in algae, that there was a thalloid organization. Over here, we are going to use the term cell aggregate, where we'll be having a cellular arrangement in which the animal cells would be arranged in such a pattern that the entire organism would be one entity, and all those cells would be working together at an individual level. Alright. Now, when I say this cellular level, I need to make it clear to you that take your own example, take any animal's example, considering dog's example or our own example. We have that animal that is having organs, then it is having basically organ system. If we talk about its digestive system, then it is having organs inside that system. Those organs are made up of tissues, and those tissues are what? They are aggregates of cells which are working together for one function. That means a group of cells doing one particular function is known as a tissue. Over here, we do not have tissue level organization. What is the reason for that? That individual cell is working as an entity for the organism. Now, when I say that this is one sponge, as you can see over here, I have listed some examples. This is one sponge. What it is going to have inside it is that it will have all the cells arranged in such a manner that this entire entity looks like one organism, and it is one organism, not just it looks like, it is actually an animal, but that animal has cell aggregate plan. So, we have cellular level. The multicellular organism is having cells which are working individually, not under a tissue name, you can say. They are working individually for a common cause. Okay, the common cause would not put them in the category of tissues. Then they are asymmetrical. So, we are done with the grade of organization, we are done with the symmetry. This is the only phylum that is asymmetrical. Take a sponge, cut it, and you will not find any point of dissection, or you can say any plane of dissection that would divide the organism into two equal parts. Most of them, they are asymmetrical. They are going to be found in water, they are marine, and there is cellular level, we have talked about it. Then we have water canal system. Before we go into water canal system, let us see the few examples that are here. One is spongilla, other is leucosolenia, we have euplectella, we have scypha. These are the sponges, they are marine, few of them are freshwater, and few of them are, most of them are marine. We have freshwater sponge known as spongilla, we have euspongia, we have the simplest sponge that we study, that is cycon, and leucosolenia, you will be surprised to know that it is a sort of best example to study the sponges, because when we study sponge, we have to deal with few things. One is that they are going to have spicules on their body. Okay, these spicules are otherwise known as spongin fibers, and you have to remember one thing, that they are siliceous in nature. Alright, that means they are made up of silica. Now, something that is made up of sand sort of substance. Now, what we have to see over here is that the skeleton has been talked about. This is the skeleton. You can say it is making the sponge hard. Now, what you see over here is the basic arrangement of the sponge. We will see about it. We are going to deal next with water canal system, that I said that we will discuss. But before discussing the water canal system, we are going to take the example of leucosolania, the best example that can make us understand the general characteristics of a sponge. What you see over here is that there are many pores which are present, termed as ostium. Singular is ostium, plural is ostia. So, we have ostia, those are the pores, and we have osculum. Now, this osculum is the mouth, you can say. Now, it is not mouth, first thing, but we can term it as mouth. Why it is not mouth? Because mouth is an organ. And we do not have tissues over here. How can we have organ for this Phylum Porifera? So, this is basically an opening for the cellular arrangement, and the pores are also an opening. So, what you are going to find is that the pores open up into a cavity known as spongocoel. When I use the term cavity, don't get confused with coelom, first thing. Second thing is that whenever I am saying cavity, it refers to an empty space or a hollow space, I would say that. Supposedly you are holding a water bottle in your hand. So, that would be the example, that an internal hollow area is the cavity. Same is in the case of sponge. That is known as spongocoel. We use the word C-O-E-L, seal, for cavity, and the term is spongocoel. There is spongocoel and there is canal system. This arrangement of sponge is such which enables the sponge to carry out its activities like digestion, nutrition. It is responsible for respiration. It is this canal system. So, let us see what is this canal system all about. We are going to see this one. Now, over here, as you can see, there are epithelial cells, okay, the outermost cells. Before going to epithelial cells, see this one. This is just like the bottle example I gave you, but your bottle is going to have pores inside it. Pores are termed as ostia. Ostial pores are present. Now, what happens is there is a water canal system. These pores are meant for taking inside water current. The water current will come and it will enter the spongocoel. See, this is the part of sponge that is being shown if we have to see its cross section. Now, see this, there are spicules which are present, there are spongin fibers which are making its exoskeleton. This exoskeleton is not to be confused with the higher animals' exoskeleton. This is the simple skeletal arrangement, we can say. We have the epithelial cells. In the epithelial cells, there are embedded spicules. Then you see that there is layer of cells which is forming the boundary of the organism. Basically, the animal's boundary is the actual animal. So, that boundary is made up of few layer of cells, the outermost being the epithelial cells. Then we have a few amoeboid cells which are present, and these green colored cells that you see over here, they are choanocytes. The term is collar cells, yes. Here I have mentioned it. They are choanocytes, the collar cells which help in the movement of whatever part, whatever water, whatever fluid that has entered into the spongocoel. This particular cavity which has taken in the water current that has come, that movement of that water current towards the opening known as osculum is carried out by these collar cells. Now, what you have to remember is that the characteristic feature of these sponges, first one is the spicules, okay, second one are the collar cells. If you are being asked about cells, these pore cells, ostia, not ostia. These are the pore cells which surround ostia, because ostia is the opening. Basically, it is the pore. So, we have pore cells which are surrounding the ostia, we have spicules, we have amoeboid cells, we have the water flow entering, water current that is entering the system. There is a gradient that is maintained so that water enters inside it. That is why the water enters through ostia instead of osculum. Otherwise, the gradient will not be maintained. That is a basic understanding of physical processes. We have the water flow that goes inside. This osculum throws out whatever water has entered into the spongocoel. Now, when this is thrown out, the other activities which are to be performed by the organism, by the cells which line this spongocoel. Now what you see over here is that collar cells, as I told you, they are responsible for moving. Not just moving, they also take up whatever is required from the water current. So for meeting the requirements, physiological requirements that the cells would perform, this particular water current is passing through the spongocoel, and the cells which are lining the spongocoel, they are going to take up whatever requirement they have, and from the water current it would be taken by the cells, and it could be respiration, it could be nutrition that is being taken care of by this water current in the cells. And you see in this diagram a small bud. When I come to small bud, that means next thing that I am going to consider is the reproduction, how the reproduction takes place. First of all, we are going to deal with this term, hermaphrodite. Whenever I say hermaphrodite, it is you can say this is synonymous with another term that is monoecious, okay, or with another term that is bisexual. Now in this case you are going to use hermaphrodite only, you will not use monoecious and bisexual, but you need to know each term individually because you haven't read the reproduction chapter till now. You need to know what it is all about. Hermaphrodite refers to a condition when both the sex organs are present within the same body. Alright, that the male gametes and the female gametes would be formed in the same body, and to be very, very vernacular in putting this thing, it would be that sexes are not separate. If the sexes are not separate, how can you see that a male or female sponge would be there? So they do not have sexual dimorphism as well. What is sexual dimorphism? We'll come to that when some organism we are discussing, it will have sexual dimorphism. For now what we have to discuss is what is hermaphrodite. It would be that animal which does not have separate sexes, and with that I mean the same organism would house the female sex organ and the same one would be having the male sex organs as well. The other term for this is monoecious, which is specifically used in the case of plants. Now what is monoecious condition? Break this word, it will be easier for you. Oecium basically refers to a condition of house, okay. Oecium is house. This is the best way. What is oecium? Oecium is house. Whenever I say monoecious, that means a single house for both male and female. So monoecious condition would be when the both male and female sex organs are present in the same body. Bisexual, that means two sex conditions are present in same organ. Do keep in mind these two terms. Okay, moving further, coming back to the actual topic that we are dealing with, that is the reproduction in sponges. They are hermaphrodites. That means within this part you are going to see some cell would act as the site of male gamete formation and some would act as the site of female gamete formation, and the sexual reproduction takes place by both male and female gamete coming in close proximity in the water system only and then giving rise to a zygote. Talk about asexual reproduction, we did talk about bud, we saw a bud coming out of it. The fragmentation method is also used. And the last one is that is the development direct or indirect? Now what does that mean? See, when we deal with development, there are two types of development. I am clearing it up. See, the development of an organism is from its stage of zygote, then embryo development, then there is formation of larva, if there is formation of larva or not, and then adult. That are the various stages of development, and that is what we study in development. Development is of two types. One is direct development and other is indirect development. Now what is direct development and indirect development? You can make it out. Direct development is that from the earlier stages of life, which could be embryonic, the organism will be similar to what it looks like in the case of adult. Take your own example. The way you were in the childhood, now also you look like that only, with minor differences that come according to the development processes which are taking place. But in the case of indirect development, I would give you the classic example of frog. Frog's larva is tadpole. It undergoes metamorphosis and it changes completely. There is no close relationship in terms of morphology between a frog and a tadpole, but the same tadpole, it gets converted to the frog. Talk about the caterpillar. Caterpillar is slimy insect sort of thing that gets quite irritating it is, and it gets onto my nerves actually. So that caterpillar would be transforming into a very good looking butterfly. So there is no close proximity again between the two, because the development is indirect. There are different stages that the animal has gone through, and they do not have the resemblance with the final form of the animal that is being formed, that is the adult stage. So that development is known as indirect development. Now coming back to our sponges from where we started, whether these sponges have a direct development or indirect development. They have indirect development. As you can see, I have mentioned larvae. Yes, they are present. There are different types of larvae that could be present. You have to remember two names. One is Amphiblastula and other is Rhagon. Amphiblastula you have to remember because we have seen the example of Leucoselenia. Amphiblastula larva is that of Leucoselenia, and Rhagon is the larval stage that is seen in higher sponges. So I guess this is enough for understanding how the sponges they fall into the category of Phylum Porifera. And from from understanding the example of Leucoselenia, what all is present inside it, you are very well aware now of the characteristic features of Phylum Porifera. So supposedly next time you are seeing an organism, you see such arrangement, do feel free to put it under Phylum Porifera and give it whatever name you want to, because still we are dealing with the diversity of life. It is ultimately all these things are going to point towards a stage where we are classifying the organism. So we start with the first classification of the animal kingdom, that is Phylum Porifera. Quite simple. I hope it's clear to you now.